Um, Good evening, everyone, and welcome. It's a real honor to be here tonight to introduce my dear friend Mones and celebrate the launch of his incredible new book, Unraveling Cancer, a book that offers a groundbreaking evolutionary perspective on cancer in an easy to understand language. It's a book that Dr. Abu Asab felt he needed to write to share his insights about cancer after a long career in the field. I've known Dr. Abu Asab for a long time. We often talk and discuss various topics, such as science, history, medicine, and of course, politics. He's an open-minded person and welcomes intellectual challenges. And I always look forward to talking to Mones, since these thoughtful conversations make us both grow and become better individuals. I've always been amazed by his passion for science and writing. Mones has poured his heart and soul into this book, and you can feel that on every page. Before we start, please allow me to briefly tell you more about Mones. He was born in Jerusalem in 1959, and he completed his primary education in Ramallah and his undergraduate studies at Ain Shams University in Cairo. He received a doctorate and master's degrees in phylogenetic systematics from Ohio University. And after a long career at the National Institutes of Health, he founded Phylo-Oncology, LLC, in 2023, a company that develops cancer diagnosis and early detection technology. Before that, he served as an ultrastructural scientist at the National Eye Institute from 2011 to 2024. And before joining the National Eye Institute, he worked at the Laboratory of Pathology at the National Cancer Institute. Additionally, he has taught at Birzeit University in Palestine. He also worked on phylogenetic analysis at the Smithsonian Institution and vaccine analysis at Walter Reed Army Institute of Research and ultrastructural pathology at the Department of Pathology of George Washington University. His record of publication is diverse and includes papers and book chapters on ultrastructural pathology, bioinformatics, tumor biology, systematics, virology, traditional medical systems, and global warming. His 2001 publication on global warming prompted the advancement by a week of the famous Cherry Blossom Festival in Washington, D.C. He's also the author of several books, including Avicenna's Medicine, Avicenna's Single Drugs, which I both personally enjoyed reading and I highly recommend for those of you interested in the history of medicine and alternative approaches to health and well-being. For the past 2 decades, he has been advocating the application of phylogenetic analysis to the high-throughput clinical data in biomedical research. His analysis has shown that phylogenetic algorithms are multidimensional tools that can be utilized for cancer modeling, genotype profiling and subtyping, as well as diagnosis and early detection. Finally, it is important for you to know that Mones is blessed by a lovely, kind wife, Nadia. She gives him support and encourages him all the time. Not to mention, she's a superb cook, which I was lucky to try her delicious meals many times, and which I personally attribute to Mones's cognitive abilities. I am so excited for you all to be here to directly hear from Mones. My dear friend, Dr. Abu Asab, please join me in welcoming him.
Thank you, Musa. Thank you very much, Musa.
Yeah.
You can always rely on Musa to make you look good. He's a kind man, very kind. I guess you want me to sit down to be able to photograph Suhaila? You want me to sit down to stay seated? Okay. I'll try. Yeah, I'll try. Talking from the table, and we'll see. Okay. Thank you for coming out this evening to hear my talk about the new book. And many thanks to Suhaila for organizing the evening. She is the one who encouraged me to do this book launch. And Suhaila is well known now in the community for doing a lot of activities. So join her WhatsApp group or look at the Facebook, and you'll see many of the nice activities that she has been organizing for our community. Also, I would like to start by saluting our people in Gaza. They have been enduring uh, atrocious genocidal war, and our thoughts with them. Please remember them, and if you can help, please do. They need every help we can give them. Uh, my journey with cancer research started in 1997, actually, when I started at the lab of pathology at George Washington University Medical School. I started in a small section there called the ultrastructural pathology section, and it is uh, a lab where you use the electron microscope to look at tissue, to look at cells. It shows you what's inside the cells. I was there for a year, and then I moved to NIH, to the National Cancer Institute, where I also worked at the ultrastructural pathology section. So it was like a direct, it's like a transfer to the same kind of work, looking at tumors with pathologists and studying. Basically, there were some projects, research projects as well. So it was clinical diagnosis and research projects, which is a nice combination, because you get to see a lot of the types of cancers that are there, and at the same time you get to do research projects with some PIs and to know more from the research aspect. So it was a really nice combination for me. I really enjoyed it. And then I moved to the National Eye Institute, where I continued to do ultrastructural work, but less of tumors. There was, the eye has several types of tumors. You can get lymphomas in the eyes, and glioblastoma, uh, and so it wasn't all cancer work at the Eye Institute, but it was a lot of studying tissues and studying processes like inflammation and studying infections. So my background has been a wide range of pathologies, which in a way uh, helped me develop some ideas about the pathology in general, and cancer in particular. So my observation after a few years of doing the work, when I was still at NCI, is that there were major components missing in the field, in the cancer field. Okay, for example, there was no conceptual framework for what cancer is. Okay, so everybody was doing research, like a shotgun approach, rather than within a particular frame, intellectual conceptual framework. And also, what was missing, the evolutionary aspect of cancer. And if you look at the subtitle of the book, an evolutionary conceptual framework. Okay, so these two aspects of cancer were not there. And as I will explain to you, you cannot understand cancer without actually invoking evolution. One of the most famous sentences in evolution is that nothing makes sense in biology except in light of evolution. Uh, One of the scientists, the population biologist, Theodore Dobzhansky, said that long time ago. And so in medicine, unfortunately, evolution is absent. It's taught little bit in medical schools, but it is not integrated in the thinking about solving the issues of cancer, or thinking about the research in particular, how evolution is important to really designing the experiments. So I noticed that those two aspects, the lack of disease concept and the lack of evolutionary thinking, were major drawbacks to understanding cancer. Uh, and to show you how much confusion there is in cancer, there are about 8 different theories on cancer initiation. Okay, so out there, when you try to understand how cancer arises, okay, it's so confusing for anybody who will try to understand. There is the genetic theory, there is the epigenetic theory, there is the genomic theory, there is also the question whether the cancer arises in cells, normal cells, or stem cells. So all that confusion makes it hard to understand how really cancer really arises. And and the confusion is even among people who you think they are like big in the field. These are the leaders of the field, and you go to lecture, and you can see the doubt in the tone of the speaker. We don't know exactly if it's only genetic or if it's only epigenetic. And then the genomic theory comes out, and all of this happened within the last 25-30 years. So you have this cascade of different thoughts and different ideas about how cancer is. The same thing about aging. There are different theories about aging. So can you imagine, with all the data out there, we cannot resolve simple issue, like, not simple, but an issue like cancer or aging. So I attribute that to the lack of a disease concept. Okay, Western modern medicine does not have a disease concept, and that's a big problem. Why? Because it relies only on apparent symptoms. Anything before the symptoms is not considered. So this is why the diseases are defined by their symptoms. But the pathology starts before the symptoms. And I'm gonna give you an idea. I mean, this thing about disease concept is not new. If you look at the traditional Chinese medicine, or Ayurveda, or the Arabic traditional tib system, they all have disease concept. For example, if you look at Avicenna's book, the first book of the canon, the disease is defined by two things, okay? The temperament and the humors. So what is the temperament? It is basically the homeostasis. See, the terminology, the old terminology is different than our modern terminology. The temperature of the tissue, our tissues, you know, our body temperature is generally around 37, but the organs have different temperatures. Like the liver is higher than 37, the brain is lower than 37, etc. So they define in the traditional medicine the homeostasis by the temperature of the organ and the humidity of the organ, because also humidity is considered a part of the health. You have to have moisture. Some organs are moist. The heart is moist, the liver is moist, the bone is dry, etc. Uh, and the other thing is the humors. The humors are really misunderstood in the Western world. Everybody tells you, "Oh, we don't know what they are exactly." No, but they are simple. They are the proteins, the lipids, okay, the unassimilated proteins, and the acids. The acids like uric acid, uh, like lactic acid, the the DNA, etc. So, all these four, there are four types of humors. So the disease in the Arabic tib system, okay, is when there is distemperament. The temperature goes up or down. The moisture goes up or down, above or below the normal homeostasis, the normal range that is supposed to be. The humors, the accumulation of a humors, okay, like if there is excess of lipid, like we know glioblastoma is notorious for accumulating a lot of lipids, okay, that's considered the chloric, as they call it in English, the chloric humor, which is the lipid fraction that goes out of whack. Uh, so this is before any symptoms is there, you know. Uh, and the other thing, like they defined uh, cancer, okay, and you find this actually in not just in the traditional tip system, but you find it also in the Ayurvedic system. Uh, the tumors produce lactic acid, so they consider the lactic acid as black bile, okay, and the solid tumors are always lower in temperature than normal tissue, okay. So they are cold and they have black bile, the lactic acid, and this is 1,000 years ago, okay. You know who mentioned lactic acid 1,000 years later, that the tumors produce that? You know it, Larry, probably. Warburg, Otto Warburg, a German scientist. He's known now for the Warburg effect, okay, which is that the cancer cells produce lactic acid, okay. Imagine they knew that over 1,000 years ago, because it is actually even in the ancient Egyptian uh, papyrus, okay. And in the book, the first chapter is actually about comparing the different uh, traditional systems and how they characterize the tumors. This is from an Egyptian papyrus called the Edwin papyrus, and this is a cancer case of a man who has tumors on the chest, and the physician on the breast, and the ancient Egyptian physician, we're talking about here about 3,000 years old, okay, touched the tumors and wrote in this case they were cold, okay. So the characterization of the tumors as being cold has been around for a long time, which will later I will tell you why they are cold, why they it's amazing. There were no, of course, biochemistry at that time, no molecular biology, but the observation was really superb, that they could tell that there is lactic acid from the tumors. Actually, they attributed the cancer formation to the lactic acid. They did not call it lactic acid, they called it black bile. But actually, we know now that it is a lactic acid. Okay, they lumped all the acids into black bile. Uh, so to see how the concept of a black bile is interesting. It's acidic, okay, which we know now that what's the composition: uric acid, lactic acid, all of those are all acidic. So as I said, Otto Warburg, I wondered sometimes if he knew some of the Egyptian writing or some of the Arabic writings, because he is the first one out of the blue to come out and say, oh, the cancer produces lactic acid. The man got the Nobel Prize in medicine twice for this work that he has done. And he was a biochemist, so he did a lot of work later, and he's characterized the Krebs cycle in the mitochondria. So the man deserves the Nobel Prize twice, actually. For now, his observation, they call it the Warburg effect. Everybody talks about it. Now it's back on. So the things that ancient physicians knew more than 1,000 years ago, now we are rediscovering it. This is to show you how much there is a disregard to nice ancient concepts. Uh, so to understand why there is cancer, we have to go back in history about 2.5 billion years ago, when the surface of the earth, or the earth itself, did not have oxygen. During 90% of planet history, there was no oxygen. Okay? The ancient gases came out of the earth, actually. This is the source of and there was no oxygen. There were compounds that have oxygen, like the carbon dioxide, the water, the sulfur dioxide. Okay. And at that time, the living organisms on earth were single cell prokaryotes, bacteria. Okay, they were not utilizing oxygen. There was no oxygen. So these organisms were living in an anaerobic environment. Okay, so the earth cooled down more, etc., and a new form of bacteria, we call it the cyanobacteria, evolved using the sunlight to break down the water into hydrogen and oxygen. And the oxygen, went into the atmosphere. Till now, the plants do that. The oxygen goes into the atmosphere, and they utilize the electron from the hydrogen, okay, into creating ATP, and they use the ATP to reduce carbon dioxide and make carbohydrates. Okay, so now we have oxygen started to filling the atmosphere of the earth. Now we have about 20% of the atmosphere in oxygen. With this, making a lot of carbon, fixing a lot of carbon and making carbohydrates, another organism evolved. See, it's a niche, because if you have all this, energy-rich compounds in the ocean, okay, something is going to evolve to use it. It's a niche. And a new bacteria called alpha proteobacteria evolved utilizing the oxygen, okay, to eat this food that the anaerobic bacteria was producing from splitting the water molecule. So now we have two kinds of organisms. We have one aerobic, using the oxygen, and the other anaerobic. Okay? So the aerobic attached itself to the anaerobic that produces the carbohydrates. And with using the oxygen as electron acceptor, managed to break down the carbohydrates into carbon dioxide, water, and energy as ATP. Okay? So basically, it was cleaning the environment around the anaerobic bacteria. The waste product of the anaerobic bacteria is life-limiting. The bacteria cannot grow in that mess. So here is the nice friendship between the two. So the anaerobic bacteria and the aerobic bacteria develop this symbiosis. Later, okay, especially with the ozone, the oxygen, plenty of oxygen made the ozone layer, protected the life on Earth. Okay, this new symbiotic entity was able to be in more than one place. Okay, but this entity, okay, is the basis for our eukaryotic cells. We are made of the eukaryotic cells. Okay, our bodies are made of the symbiotic system, the nucleus, okay, from the anaerobic bacteria, and the mitochondria from the aerobic bacteria. Beautiful system. You have now a cell with its energy generating, okay, uh, organelle, okay, because now we have a cell. We don't have two bacteria distinct, like the mitochondria cannot say, okay, goodbye, I'm getting out. No, now they have a system that gave us the eukaryotic cell, which has a lot of advantages over the prokaryote. Okay, so now with plenty of energy, you can have bigger cells, you can have more chromosomes, larger nucleus, you can have more membranes, you can do differentiation, etc. You can have multicellularity. Before the prokaryote, you could not have a multicellularity. So the eukaryotic cell, okay, gave a new form of life that produced, and remember, most of the anaerobic bacteria was in the ocean, all right, till the oxygen filled the atmosphere. So now you have new entities that are capable of moving to the surface of the earth and live anywhere they want. They have their, powerhouses with them, and they can differentiate. They can have multicellular organism. So this is the basis for the adaptive radiation, what we call it adaptive radiation. All the new organisms that started filling all the niches on Earth. Okay, so what does this have to do with cancer? You are gonna ask me. Okay, so during my 26 years looking at tumors, all tumors have dysfunctional mitochondria. Imagine you have a cell, healthy cell, spending 50% of its energy. Okay, the mitochondria produces 90% of the energy. So producing 50% of its energy on maintenance and the other 50% on differentiation. Now, the energy in the cell went from 100% down to 10%. So what's going to happen? There is no differentiation anymore. This is what cancer is. No differentiation. It loses the differentiation. Okay? There is no energy to do anything. The cell does not even have enough energy to kill itself. This is why cancer cells do not die. Apoptosis, which is the programmed cell death, it's an energy intensive process. For the cell to kill itself, it needs energy to digest the DNA, to digest the membranes, etc., and get done, basically. But the cancer cannot do that. Why? There is no energy. So the cell reverts to a bacterial-like state, okay, that we call cancer. So now you see how evolution helped us solve why cancer does not have enough energy, why cancer even starts. It's the lack of energy from the mitochondria, because all the mitochondria are dysfunctional. And I put pictures in the lots of pictures from the electron microscope, and you can see, like in this one, where the mitochondria have lost their integrity. The inner membranes, which are usually beautiful, crysty, etc., they are like a soup. Okay, so this is why solid tumors are cold. There isn't energy to keep them warm. And Uh, that's in a nutshell how cancer starts. Cancer starts by having a hypoxic phenotype. Lack of oxygen, okay, will cause also the transformation. So it doesn't have to be a mutagen to cause cancer. It can be as simple as hypoxia. Yeah, some region in the body does not, like let's say kidney, etc., does not get enough oxygen. It will develop cancer. So oxygenation is important. We talk, Musa and I, we talked about this a lot because he is a yoga master, and and we talk about it because we got to the conclusion that yoga is about circulation and oxygenation. That's what it is basically. It's not about the physical exercise per se. No, it's about oxygenation and circulation. So the disease concept that I'm advocating is that it is the uh, uncoupling between the nucleus and the mitochondria that takes place that triggers the cancer. There has to be either partial or entire coupling that takes place in the cell to become a cancerous cell. And we see that by electron microscopy. We see that. We see how the, uh, the mitochondria in the cancer cell are totally dysfunctional. And actually, this thing when I was at the National Cancer Institute, I noticed it because all the pathologists, when writing their reports, they never talk about the mitochondria, as the mitochondria does not exist. But the mitochondria is the most important part. The mitochondria is the part of the cell that actually keeps it healthy, keeps it differentiated. Differentiation here is a key. Most of the what differentiation means for those of you who are not physicians or scientists. Differentiation is the primary function of the cell, like a liver cell has its function. So 50% of the energy of the liver cell goes toward doing the detoxification and the digestion, etc. The same with the kidney cells. They have to have differentiation into tubules to function. So if you don't have enough energy, there is no differentiation. So, but let's suppose there are some mitochondria, and not all the mitochondria died or got dysfunction the way in cancer. Then you get a pathology. Most of the pathology is that when the organ, okay, does not have enough energy to function. So, so this is actually the energy-centered, okay, disease concept, that you have to have enough energy for health. If you don't have it, and if the cell does not have it, the cell becomes diseased, okay, the organ becomes diseased. And believe it or not, Avicenna stated that 1,000 years ago in this book, actually. And actually, I re-put it again in this book. So you have it about the energy. So pathology, in my opinion, okay, is energy based. Okay, so without having enough energy, there is the cellular problems, and this is what you get. The diseases that you get actually start in the mitochondria not producing enough energy for the cell to keep its differentiation. Uh, so this is the new. Uh, I mean, I mean it's new and not new. I cannot claim. I reached it independently from Avicenna. But then I found that he mentioned it. So we have to give the man a credit, for talking about the energy-centered disease concept. So, I mean, I'm happy that he mentioned that a long time ago. At least we know that somebody was smart enough to reach it without all the tools and instruments. I mean, I had to use an electron microscope to figure it out. I don't know how he figured it out, but he figured it out. He was. I mean, these concepts were there. These are ancient concepts. These are not new concepts, and you find them. You find, for example, we were talking about lactic acid and the accumulation of humors. Okay, these are ancient ones, and the first mention of the black bile, the acids in disease, actually in the Egyptian literature, the ancient Egyptian literature, and the Greeks did not have the black bile. They borrowed it from the Egyptian. Thales, who the Greek claims him as one of their great physicians, actually was not Greek. He was Syrian, and he's the one who studied in Egypt and introduced the black bile to the Greek medicine. So basically, the Greek medicine is not Greek. It's it comes from ancient, Egypt. Anyway, but that's not what I wanted to say. So so again, this is to show you how when you invoke evolution into the scientific literature, you get to resolve many of the things that are hard to resolve. Okay, and and I think one of the missing aspects, like in training physicians, is that to train them in evolution, you have to attribute many things you can attribute it to evolution. And without doing that in medicine, you cannot solve many issues. And this is why I think that the evolution, let's say evolutionary medicine, probably we should call it, Larry, evolutionary medicine, should be into. Actually, there is a journal now, the evolutionary medicine. There is one that came out a few years ago. Uh, so to add more to the story, there are also other primordial issues that we see in the cancer that nobody attributes it to mitochondria or other things, which is one of them is autophagy. Autophagy is a process where small vesicles are produced inside the cells. Okay. And my own research, and I was the first to publish on this early in the 2000s. I think 2003, I published an abstract at the American Cancer Society, uh, where I showed that it was the mitochondria producing those small vesicles. Okay, and it's a field now. The autophagy research is huge, but what I showed in that abstract is that those vesicles, they actually contain the chemotherapy. If you give chemotherapy to the cancer cells, one of the things that they would do, they will sequester the therapy inside those vesicles. Okay, and you can see by electron microscopy that those vesicles, the autophagosomes, they come out of the mitochondria and they travel in the cytoplasm and they go to the membrane and they fuse with the membrane, dumping out the chemotherapy. I, I put few images of that also in the book to show how the autophagosomes, they contribute to chemoresistance. Uh, and this is you see those vesicles not just in cancer cells, also in the tubule cells in the kidney. When there is septicemia, those mitochondria inside the tubules, they'll start sequestering also the toxin and dumping it out. And actually, we did this work with a group at NIH where we took the urine and we spun it down and we got the vesicles in the bottom of the tube, and when analyzing it, we found that the toxin is there. So it's an ancient, it's a primordial way by which the mitochondria used to sequester toxins and dump them out. Okay? That aspect, that evolutionary aspect of the autophagy research is not there. Everybody talks about this gene, that gene, etc., but the complete story of why there is autophagy, it's not there. The other aspect of cancer that I like to talk about is the heterogeneity of cancer. Cancerous cells are heterogeneous. What does that mean? Let's say we have a tumor and we fractionate this tumor into its cells, individual cells. And I want to measure the expression of a gene in each cell of those. You'll find that the level of expression in each cell is different than the other. It's not the same. Every cell has its mixture, okay, of expressions that are dissimilar to other ones. And why? Because this is a process that you find it in populations in nature. Like let's say the coronavirus. Let's say the coronavirus spread all over the world. It's not going to kill everybody. Why? Because we are different. There's going to be a group that are resistant. The same with cancer. Okay, if you hit cancer with chemotherapy, with radiation, there's going to be a fraction. Even if it's less than 1%, you're going to see a fraction that's going to survive. And we see that, like treatments, you see that. Why? Because the heterogeneity produces so much variation, and certain combination is going to be resistant, the way humans are. There is no virus that's going to kill humanity. There is going to be group that is resistant. So having said that about heterogeneity, so then you ask, how the heck we are going to analyze the data, how we are going to figure out what's going on. Well, you have to use a method that is compatible with heterogeneity. And heterogeneity is an evolutionary thing. So in the old days, because I'm Mr. Evolution, I looked for what is compatible with heterogeneity data. And I had worked in the 80s, believe it or not, on phylogenetics. Phylogenetics is a field of analysis, of data analysis. And it's an evolutionary field. Basically, it was built to show you which species is derived from what species, the relatedness, the evolutionary relatedness, okay? So it has built in it a way to deal with heterogeneity. And when we applied phylogenetic analysis, okay, to cancer data, it gave us nice results. If you have enough specimens, okay, healthy primary tumors, metastases, and you analyze them by phylogenetics, you get a cancer spectrum. What is the cancer spectrum? You get the healthy, you get the primary, you get the metastases, all lined, on a line. And even between the healthy and the primary, you have some of the healthy who are accumulating, okay, changes falling in between the healthy and the primary. So the phylogenetics gave us a true cancer spectrum. It really successfully dealt with the heterogeneity of the cancer data. And actually, we patented this in 2007. And now we have a company with my friend Mohammed Shawshi that you all know to try to commercialize it. Why? because this is the earliest detection method for cancer. It basically detects the early changes that are taking place in the metabolites or in the proteomics of the not even you, the analysis is based on the serum. It's not based on the tumor. No. We are way before tumor. Okay. So even the early changes that are taking place inside a person, if they are developing cancer, if they are going to develop cancer, you can detect it. You can detect it in the serum of that person, not from digging out cancer, free DNA. No, this is this is based on the uh, the different the changes in that is the tumor creating in the serum. Okay, it looks like magic, okay, but it's real. And the last chapter, chapter 12 in the book, several examples of analyzing that and doing that. This is the cladogram. This is what the phylogenetics, like here is the spectrum, the cancer spectrum, and below it is the tree, the phylogenetic tree that tells us, how specimens are related to each other. It's a beautiful method. I mean the way I have it in the book, it's simplified. So dig into it and learn about how we do that.
So you use it even like for annual physical and
Yeah, you can do this test as an the annual.
this is great.
Yeah. This is
Especially for people
I tell Muhammad, my partner, this is my claim to fame. I mean, that was my best bullet.
well, hurry up.
Well, hopefully before I die.
This is amazing.
Okay. Yeah. Well, I'm done. So anybody who wants.
So follow up on that, you're saying it's a screening test. And what is different between that screening test and what we call what pathology, as you mentioned, DNA in the serum?
Yeah.
Where do you can you expand on that?
Okay. So this data that I have in the book, it's based on gene expression from tumors. It is based on proteomics from the serum of the patients. It's based on the metabolomics also from the serum. Okay. So so basically you have to take a blood draw and take the serum, do digestion on it, and then you stick it into a mass spec machine. Usually it's a laser beam that hits the specimen, and you measure, the time of flight for the different compounds, etc. So it's a wet test, but it's a minimally invasive test.
So what is specificity? What is this? A negative predictive value? What is how accurate is it? Have you done
Okay.
that on the studies?
Okay. Yeah. For as you can see in the figures of the book, we do not have a misdiagnosed specimens. Okay. The reason, you have to understand phylogenetics. Actually, phylogenetics is not a statistical test. It's not a machine learning. Musa can tell you about machine learning. That's his specialty. It's basically taking the specimen, and based on the values that we get from the machine, it sticks it on the tree where it belongs. Okay, so the tree is a summary of the matrix. So what determines where you are on the tree is basically uh, how much derived similarities. So aberrations, how many aberrations you share with this specimen or with that specimen, or lack of aberrations. So it's a I can explain it more to you. If you
So I guess my question is not that. My question is, let's take 100 people. Take 100 people.
100 people.
What is the chance of having false positive data?
Uh-huh.
What's the chance of having false negative data? That's
Y-
the question that people ask.
Okay.
Before you even
Okay. Okay. Yeah. And you see, you're talking about statistical.
Absolutely.
Okay. And the phylogenetics, okay, there is no, there are no mistakes in it. So if a specimen is cancerous, it's gonna be lumped with the cancerous clade group on a branch, okay, depending on its profile.
There's no false positive.
No false positive.
And then the question comes in, if it's cancerous, then you're trying to figure out if it's benign, metastatic, or benign, wrong term. If it's localized, metastatic, where it's where it is.
Where its location on the tree of cancer.
Would you combine that with a wet pathology? For example, somebody says, okay, you have adenocarcinoma, you got prostate cancer.
Uh-huh.
And now with this, I can tell you it's just not adenocarcinoma. Adenocarcinoma is metastatic. It gives you more information in combination with other studies.
Mm-hmm. It's basically, we done only 2 groups of cancers together, and they still separated. So what I'm hoping one day, when we have the funds and we have the time to do it, is to build the tree of cancer, where we can know the relationship of each type of cancer, what it is. So you give me a specimen, and I will run it, and we know exactly what type of cancer, by putting it with the tree of cancer. Yeah, it is a novel method. I mean, it's not novel. I mean, this method has been there in the 70s, but we were the first to apply it to cancer. Okay, I'll tell you the story. We had interesting story at NIH. The Congress allocated about $50 million to do clinical proteomics. Okay, and that is to take the serum proteome and see if we can separate the specimens based on their proteomics. Remember, Lance Leota was doing this work, and the method they had the data, but they did not have the method to analyze it. Okay, so they hired a company, Corilogic at that time. They hired Corilogic to do the analysis. So Corilogic gave them, like 4 groups based on the profile of their numbers. Okay. When they applied the same algorithm, the same method, okay, to a different cancer, they started with pancreatic or prostate, and then they moved to pancreatic. They did not get good results. So our chief at that time, Lance Leota, was explaining this, and this is when it occurred to me, why to create different algorithms for different cancers? We can use phylogenetics, okay, and we don't have to do that. So I went to him. I knew him personally. This guy had a lab of 30 people working on this stuff. He was invited every week to give a talk somewhere in the world, not in America only. So I went to him and I said, you know what? We don't need different algorithms. We can use phylogenetics. Of course, he had not heard of phylogenetics. So he said to me, show me. So I spent with Muhammad the next 6 months analyzing in order to show him. And we were not sure, if we had it correct at the beginning, and then we started looking harder, at the results and figure things out. And sure enough, we started seeing a pattern. So we summarized it into smaller trees, like the one we have. And sure enough, we have a method. So by the time we figured it out, the guy got into ethical troubles. They kicked him out. Yeah, he would have stolen it. He was well known to steal, ideas, and so he was gone. So now we don't have, a sugar daddy to finance the research. But we continued. We published about 10 papers different cancers. Every time we analyzed data and we found, wow, this is interesting, we wrote it up and published it. So, yeah, Dr.
And how
Amira.
did you get to patent it?
We tried to patent it through NIH. They refused.
Oh,
So we at that time, a friend of mine worked at Georgetown University, and we patented it through Georgetown. But then Georgetown, after the first year, they said, no, we are not interested. So we patented it on our own.
good.
But it took a while because it took them a year to figure out how to categorize it. Okay, and then this examiner would ask us every question under the sun. I spent hours answering his questions, but finally we got it. We have the patent. Yeah. Yes. Yeah. Yeah. Go ahead.
One last question. So you get the data analysis,
Mhm.
let's say identifies an early detection of
Yeah.
cancer,
Yeah.
but it doesn't identify where that cancer in the body is.
No. But this allows depending. You see, you look at the location of where that person fell on the tree. If it's in proximity to the primary cancer, then you treat them as a cancer patient. You give them chemotherapy. Or if I mean, probably at that time, microscopically, it's not detectable. Okay, so it's a clinical decision what you want to do with the patient at that time. But basically, you could watch and see where is that exactly. So you can observe them till you are able to. Majdi, let Dr. Amira first. Yeah, Amira.
About the staging cancer. They say this person has
Yeah,
stage one,
yeah,
two,
yeah,
three, four.
yeah.
Does that depend on the location and how critical place in the body, or they are looking at the cancer cell, how aggressive it is?
Yeah.
Because some cancer cells
Uh-huh.
be more aggressive than
Of
others.
course.
So how do they do this staging?
The staging, I'm not a clinician, so Dr. Ahmed probably could answer that better than me about the staging. The staging basically is a combination of what you said. It's looking microscopically at the cells. The more primitive the cells, the less differentiated. Okay, that they are more aggressive, and then you go with the location if it metastasized, where are the metastases, etc. Yeah.
As I understood, that the mitochondria is very important
Uh-huh.
in the cell.
Yeah.
So because of no oxygen, it would die, or then it is some blood vessels comes out of it. I'm seeing, of course.
Yeah.
But do they think about giving oxygen when they start seeing those cells deprived of
Yeah,
oxygen?
yeah. I am one of the people who believe that uh, once the cancer cell is cancerous, you cannot reverse it. It's irreversible. It cannot be reversed.
Well, I think there's bladder cancer.
This is very interesting, but I have to go.
Yeah, I mean,
Sorry.
no problem. Thanks for coming, Nada.
Dr. Mones, I just have a couple of quick points.
Okay. Yeah, go ahead, Majdi. Yeah, well, we have plenty of time. If you are gonna stay, I'm staying. I mean, Suhaila, how long do we have? We have plenty. Okay, okay.
That you cause
Yeah,
to
let-
differentiate, I think one is.
Okay.
Cancers that you cause to differentiate, and once they differentiate, then they sometimes stop being cancers. Yeah.
Larry, I did not understand. What did you?
Well,